welcome to this worldwide lab meeting from the Psychiatric Genomics Consortium. I'm Cathryn Lewis. I'm an executive director of the Psychiatric Genomics Consortium, the PGC, and co-chair of the Major Depression Disorder Working Group. And I'm Professor of Statistical Genetics at King's College London. So the worldwide lab meetings are the PGC's regular scientific forum, and they give us an online platform that we can all hear about the exciting research that's going on in psychiatric genetics, both inside and outside the PGC. And if you have ideas of what you would like to hear about, or if you would like to offer to talk, please do let me know. These sessions are open to all. Do forward the information to your colleagues and collaborators, and all recordings are placed on the PGC YouTube channel there. So today's worldwide lab meeting is a very special one. It's to celebrate a new initiative in the PGC, our new working group in functional genomics, which is led by Mary Ellen Lynall and hence Hjerling-Leffler from the Karolinska Institute. Mary Ellen's at the University of Cambridge. And today they're going to both introduce the working group and give us some scientific talks around the areas that they're working in. And then we're going to have 3 talks. So first, Mary Ellen will introduce the functional genomics working group. And then Janitza Montalvo-Ortiz from Yale will talk about her work on large-scale meta-epigenome-wide association study. And then Jens Hjerling-Leffler will talk about data-driven prioritizations of cells and circuits in schizophrenia. So without further ado, I'm going to hand over to Mary Ellen to give us an introduction to the group. And before I sign off, I just want to say we'll run all those 3 talks one after another and hold a common Q&A across all the presentations at the end. But the Q&A panel is now open, so you can add questions to that at any time during the talks. Okay, handing over to you, Mary Ellen.
Thank you very much for the introduction, Cathryn, and thank you for having us to this worldwide lab. So my name is Mary Ellen Lynall. I'm an academic psychiatrist at the University of Cambridge, and my main work focuses on the contribution of immune cells to psychiatric disorders using bioinformatic and wet lab approaches. But I'm here today to talk to you about the newly formed PGC Functional Genomics work group that we kind of have started meeting in November 2023. And a lot's happened already, so I'll whip you through our mission, our leadership, the kinds of data that we're collecting, the initial projects, and how you can get involved in this exciting new venture. So to begin with, our mission is to be a dynamic hub for collaborative open discovery science and scalable method development. We want to drive inclusive and diversive omic health research, fostering a collective effort that transcends individual capabilities. And that's really what it's about. It's about doing things together that we couldn't do alone. And as Cathryn mentioned, Jens and I are co-chairing this effort, but the structure is a little different to your standard PGC work group in that while we meet monthly in all hands way, the core work of the group is done in the project work groups led by the project leads that you can see here. And this structure is partly that, there's a huge number of different scientific questions that we could have asked within this group, but we focused on what we think could be the low-hanging fruit for our group, what data sets are available, and what the interests are of the people within the group when it formed. But of course, there'll be lots of space in due course for other projects and other questions. And we've been really lucky to be able to build on the existing momentum within the PGC for the functional genomics area. So there's, we landed on our feet with an enthusiastic group of collaborators, and it's been a huge pleasure to get to know this group of people. So what kind of data are we dealing with? Well, this is very much a cross-disorder effort, and we put out a data census asking people what kinds of data they would be able to contribute. And as you can see, this covers a whole range of different disorders, and people from across the PGC and out with the PGC have filled out this census to tell us what they will and will find less easy to share. And on that note, we have a similar challenge to the rest of the PGC in that some people are able to share individual level genotyping data, important for us for quantitative trait loci, and some people are able to share summary statistics. And of course, there will be molecular epidemiology data sets not associated with genomic data. Another important factor for us is the phenotyping information about health. And for the omic data, that's not a nice to have, it's really critical because so many socioeconomic and environmental factors affect biomarkers. And to give you a sense of the kind of data that people are able to share on that front, you can see that there'll be fairly high levels of sharing of things like smoking, yes or no, but lower levels of being able to share things like the medications that people are taking, history of infections, stress, things like that. And an additional challenge for us is the sheer diversity of data types and tissues that we'll be integrating and collating. Now, it'd probably not surprise you to learn that looking at the brain data sets on the left and the non-brain data sets on the right from our data census, the most commonly held data sets are brain RNA sequencing, and in the blood, methylation and proteomics. And those will be, that lines up with our projects, which focus on some of those data types where we have a lot of data sets to bring together. So I'll whip you through a few of the project groups, what the aims are, and do keep an ear out for which of these you might be interested in joining, because we're very much looking out for both data contributors and analysts for all of these groups. We simply have an Excel sign-up sheet that I'll ping up at the end, and you can get involved. So the first work group, led by Shen and Lawrence Nisbet at Edinburgh, is about multi-omics QTL data. So a big challenge for the field here is that if you've got a given loci of interest, locus of interest, while you might be able to see the pQTL for that, if there happens to be a significant pQTL in that region of the genome, the subthreshold signals across other omic layers, such as methylation QTLs or chromatin accessibility QTLs, you might not be able to see those because they wouldn't have been publicly released. People don't always store that information. So what Shen and Lawrence are doing are working with the data contributors to try and generate multi-layer QTLs at key loci for psychiatric disorders, such that that data can be considered and integrated across those different levels of analysis. And they're going to create a harmonized data set and a Shiny app that will be a centralized resource on Snellius that other groups will be able to use to do simple analyses like CoLock and SMR using this new data asset. I think that'll be a really nice contribution for the wider PGC community. Second work group, I'll touch on only briefly because Janitza is about to tell you a bit more about her work in this space, but this is going to be a large-scale meta-EWAS, cross-disorder, cross-tissue, characterizing the epigenomic landscape of psychiatric disorders, using both standard EWAS methods and machine learning methods, and integrating across different omic layers. So again, meeting monthly, please do sign up. Next, we have Hae-Young Won at UNC, who's going to be leading on functional validation of GWAS variants using screens such as EMPRA and CRISPR screens to identify a regulatory principle of key GWAS variants for psychiatric disorders. So she's got a postdoc starting who's going to be bringing together the existing EMPRA data sets, establishing a uniform pipeline for their analysis, and calibrating reproducibility across the different assays. From John Mill in Exeter, we've got a work group that's bringing together long-read transcript sequencing from the brain to identify patterns of alternative splicing and protein isoforms in the human brain. So looking for anyone who's got long-read sequencing to contribute data sets. This is going to be really important for identifying novel transcripts. We already know that work in this area from John and others has shown that, in fact, exome sequencing isn't going to cover the full range of transcripts that we need to know about. And this work will identify refined annotations for burden testing that other groups in the PGC will be able to use. Finally, Jens and I are leading the work group on the single cell data frame, with Jens leading the brain effort and me the immune effort. We're bringing together a centralized, standardized data frame of single cell data relevant to psychiatry in the blood and brain. So we're going to integrate, there are a huge amount of efforts in the single cell area, and we're going to focus particularly on case control data and integrate that into atlases with larger data sets from healthy participants and do a meta-analysis of case control changes because reproducibility has been another big challenge in the single cell area for other diseases. We're going to produce a central asset on Snellius of these data sets and a pipeline to facilitate functional enrichment analyses and generation of eQTLs. So we're collating this data. We're especially interested in hearing from people who can also share phenotypic data and health data on their single cell data sets. So finally, please get involved. If you'd like to be added to the mailing list to attend our meetings, which are open to anyone who's interested, contact Fatima. You can sign up for our different work groups or to give a science talk. We've had a phenomenal set of science talks to kick us off. We're interested in hearing it work at every stage of its development, 20 minutes plus Q&A. Do put your name down. And also data contributors. We'd love to hear about any other omic, QTL or EMPRA data sets that you might be able to contribute. No commitment at this stage, but if you could add your details in the quick survey, that would be fantastic. And finally, we're keen to co-produce this work as much as we can. So we'd like to involve patients and the public from early on. We've got good links to patient groups locally for ourselves, but we're keen that this be an international effort. So please do get in touch if you know patients who might be keen to help co-produce this functional genomics effort. And Jareth Wolf from Edinburgh is leading on that. So please do drop him a line. Finally, future meetings. It's a monthly online meeting, alternating early and late slots to facilitate participation worldwide. The next one is on the 20th of May. We've got a face-to-face meeting lined up in Singapore at the World Congress. We're going to have a social then as well, and we're looking forward to getting to know each other. Finally, we've been doing a sort of mini roadshow around the various different PGC working groups. We've still got a few to go, but that has been enormously helpful. We've really appreciated groups being so open and sharing what they're doing with us. And it's been really great to think with the groups that we visited so far about how we can collaborate. Because of course, this is, we're very aware that there's been a lot of functional genomic efforts going along in the consortium already. We want to integrate and collaborate along these lines. We certainly wouldn't be doing analyses about a disorder without the relevant group involved. So we're keen to work in a very cross-disorder, cross-group way. So thank you very much to the PGC for supporting the setup of this group. It's been a really exciting few months. And thanks especially to Pat Sullivan for supporting this and to Andrew McIntosh, who has been absolutely critical to the early setup of his group. And it wouldn't have happened at all without him, of course. So we're really grateful for that support. Thank you.
Thank you very much, Mary Ellen. That was a lovely overview, and I appreciate your comments, thanking Pat and Andrew as well. And a very warm welcome to the PGC. It's great to have you with us, enriching our work, and particularly nice to hear how you were talking about working with the disorder groups and also working with people with lived experience as well. That's great to hear. So, as I mentioned, we are going to move straight on to the next talk, and that's from Janitza Montalvo-Ortiz, and I can see your slides up already, your meta-epigenome-wide association study. So Janitza is at Yale, and of course the other place we know you from very well, Janitza, is all your work in the LAGC, which I'm hoping you'll cover to some extent here. The floor is now yours.
Thank you so much. So today I'll be talking about what we envision to do as part of the PGC Functional Genomics group, but more from the lens of molecular epidemiology, focusing on large-scale meta-EWAS efforts to identify biomarkers of psychiatric disorders. So why looking at epigenetics? So we know that functional genomics goes beyond help prioritizing genes identified in a GWAS and also help characterizing the function of genetic variants. Functional genomics also encompasses the interaction between our biology and environmental factors, which we know that it could be mediated and hopefully captured by epigenetic mechanisms and signatures. One of the most studied epigenetic mechanisms, as we know, particularly in humans, is DNA methylation. DNA methylation, simply put, it's basically the attachment of methyl groups to the DNA, and how they regulate gene expression is whatever methyl groups are present in the promoter region is associated with gene repression, and whereas not present is associated with gene activation. I'm going to talk about specific projects done in my lab and how the information that we have recovered or discovered, I would say, has to be considered in larger scale EWAS efforts. And we hope to implement this in our efforts at the PGC FG group. And why focusing on opioid epidemic and opioid use disorder? We know that 2.1 million people have had opioid use disorder, over 100 people dying every day, with a more than 40% increase in a 12-month count of opioid-related deaths in 2019, and representing around $78 billion of annual economic burden. What is known about epigenetics of OUD, we know that OUD is influenced by both genetic and environmental factors, and there has been quite a literature on epigenetics in this trait. Early on, of course, the research was mainly focused in targeted candidate gene studies, mainly in the OPRM1 gene, which is one of the opioid receptor, part of the opioid signaling pathways. But more recently, epigenome-wide association studies have been conducted, looking at this at the genome-wide scale. The first one being conducted by our lab, and when I was also in Joel Galanter's lab at Yale. And we were able to identify 3 differentially methylated genes, including RERE, and I'm highlighting this because it will be also shown up in later slides. And this is a very interesting gene because it has been previously identified in GWAS of schizophrenia and GWAS of using MTAG methods in depressive symptoms as well, and loneliness. And more recent EWAS studies have found alterations across the genome, and this has been looking at not only whole blood, which is also the tissue that was examined in the first EWAS, but more recently brain tissue, at least from 3 different research groups. But we need to consider that these epigenetic alterations, different from genetic variations, are cell type specific. And that's why it's key to understand what's happening in the brain whenever we want to identify biomarkers, so that we hopefully can integrate and doing more cross-tissue analysis of whatever biomarkers were identified in the blood, we know that they're brain relevant. But when looking at the brain, most of the DNA methylation studies have been using array-based methods, which doesn't really distinguish DNA methylation from other epigenetic marks like hydroxymethylation. And particularly when looking at the brain, this is key because of the high prevalence of this epigenetic mark in brain tissue. So what's hydroxymethylation? This epigenetic mark is part of the DNA demethylation pathway. So it's the first step of demethylating DNA that happens after oxidation of the methyl groups. So we know that if this is a stable epigenetic mark, that it has a big role in transcription regulation, mainly activation because of the colocalization with enhancer activity, enhancers. And it's very specific to brain tissue, highly enriched in neuronal cells, has been implicated in neuronal plasticity, and has been implicated in a wide range of disorders, including depression and now more recently substance use disorders. So in order to better understand the role of DNA methylation in the brain, and because of these a priori information about hydroxymethylation, our lab conducted the first parallel investigation of DNA methylation and hydroxymethylation, specifically in human neurons. And we did this looking at tissue from the orbitofrontal cortex, using FANS technique to isolate neuronal nuclei, which was then used for reduced representation oxidative bisulfite sequencing, which is a technique that we used to assess both methylation and hydroxymethylation and being able to distinguish between these two. This is just an overview of the coverage and performance of our sequencing assessment. We had an average of 10x coverage that obtained around 3.5 million CpG sites, which is more than 3 times higher the coverage of commercially available array. Assays are used for DNA methylation, mostly from Illumina. This the coverage was very good also in promoter regions, with 44% of CpG sites being located these regions and 30% in intergenic regions, with an average of DNA methylation at each CpG sites been 20% and for methylation and 10% for hydroxymethylation. We identified a total of 397 CpG sites significantly differentially methylated and 1,740 differentially hydroxymethylated after multiple testing correction. And as we can see here, this is a circles plot showing all the epigenetic marks identified, whether those of DNA methylation in blue and DNA hydroxymethylation in pink, and those showing hyper direction in red and hypo are decreased in green. These differential marks were mostly observed in promoter region and with most of them in hydroxymethylation marks in promoter region compared to DNA methylation. And when looking at the overlap of these two, we don't see an overlap at the CpG level, but we do see some overlap at the gene level. In terms of the functionality of these marks identified for DNA methylation, we don't see any significant signaling pathway after multiple testing correction, with some nominal significance of at least 2 related to opioid pathways and also nicotine. But in hydroxymethylation, we were able to observe significant signaling pathways, including the GP signaling pathways, the Wnt signaling, and also GABA-B receptor signaling, and with a FDR 0.05 opiate signaling as well. Looking at more gene ontologies significantly enriched with hydroxymethylation, mainly in G protein signaling, postsynaptic differentiation, GABA-B receptor signaling, and trended significance for opiate signaling. We also did looking at beyond specific CpG sites, looking at neuronal networks, doing a co-methylation analysis. We identified 10 modules for DNA methylation and 4 modules for hydroxymethylation associated with OUD. For the DNA methylation modules, these were mostly enriched for transcription regulation, cell differentiation, nervous system development, and neurogenesis. And from the 4 modules identified in hydroxymethylation, at the pathway level, we observe a similar enrichment. So integrating GWAS data with our finding in neuroepigenetics in the brain, we observed that for hydroxymethylation, for the differential analyses, multiple enrichment for GWAS of disorders including schizophrenia, sleep functions, bipolar affective disorders, temperament and personality function, and so on. and looking at the co-methylation, co-hydroxymethylation analyses from the modules, from the genes included in those modules, we do observe a lot more enrichment and a lot more enrichment observed with hydroxymethylation, including psychiatry subdomains and also those related with alcohol and tobacco. So we also did a gene-drug interaction analysis to look at the drug that interact with the genes that show differential methylation or hydroxymethylation. And we found drugs that are, these are just showing drugs related to opioid specifically, and the difference between methylation and hydroxymethylation, with hydroxymethylation identifying drugs related to opioid, but also drugs that have been treated for withdrawal symptoms from opioid use disorder. So when looking at the brain, it's also important to look at beyond CPG sites, which has been underexplored and understudied so far. And the reason why is that is because methylation at non-CPG sites, while it's usually low in peripheral somatic cells, it has a very high prevalence in embryonic cells and brain tissue, and very highly enriched in neuronal cells. Its role in gene regulation is also related to gene activation. And while I'm not going through details on our finding because of timing, you can access a paper with this QR code, and this is basically summarizing the findings from the methylation and non-CPG sites with those that I just already covered. And in summary, we're identifying a lot more marks with non-CPGs that seems to be also unique, with some of them also shared across methylation and hydroxymethylation at CPG sites. And more recently, we also performed a methylation and hydroxymethylation rescores in an attempt to potentially design biomarkers. So this is in the brain, of course, but the next step will be to do it in large-scale EWAS of opioid use disorder, and then hopefully integrate the two of them and see how they explain the variance of OUD. So for these analyses, we compared methylation of we built methylation and hydroxymethylation rescores. These are the ORC plots with the number of genes identified for methylation and hydroxymethylation, and how much, here we have how much of the variance is explained. And it's very clear that compared with methylation rescores, hydroxymethylations do explain a lot more of the variance explained, with close to 35%. And when integrated, the two, it's basically about the same as just with the hydroxymethylation. And considering covariates, the same covariates that we did with the other analyses, covariates alone do explain around 10%. Covariates with methylation rescores do increase the variance explained to up to 20. And covariates with hydroxymethylation explain around 40%. So we do see a significant effect of hydroxymethylation rescores in predicting OUD. And here's, I'm just showing the different genes that were identified in the methylation rescore that are part of the opioid signaling pathways. That could be interesting targets to and biomarkers of OUD. And also have to mention that another gene that we identified, it was RERE in the brain for methylation, which also was identified in the blood in the very first EWAS that we reported. So this highlights the need to do this, but looking at cross-tissue perspective and having more brain-informed biomarkers. So in conclusion, in the brain, we found that hydroxymethylation plays an important role in OUD and is functionally distinct from DNA methylation. For this, we observe enrichment and with signaling, G protein signaling pathways, and near significance for opioid signaling, Most of these occur in promoter regions, which suggests a role in gene regulation. Some overlaps observed at the gene level, but not at the CPG level. And also similar gene networks are targeted. We do observe enrichment for GWAS of a wide variety of psychiatry and developmental disorders. But again, more enrichment is observed with hydroxymethylation. And the same in terms of drug interaction that were identified, including drugs that are used for the treatment of OUD. And in terms of risk scores, hydroxymethylation seems to outperform methylation risk scores, and we identify genes involved in the open signaling pathways and also those identified in previous EWAS of OUD, but in whole blood. And just going briefly, what we also aim to do in the meta-EWAS group is going beyond epigenetics and looking at transcriptomics to further understand the regulatory role of epigenetic mechanisms in gene expression. And this is an example of the PGC SUD epigenetics working group, which is we have as a preprint doing a meta-analysis, transcriptome-wide analysis of opioid overdose. So if you want to learn more, you can access the paper in this code. And this is just showing the aims of our group, but I want to highlight that there are also other PGC efforts doing meta-EWAS and also integrating with other omics as well, namely PGC PTSD, which has a long track record of doing these efforts. More recently, the PGC SUD and also the PGC MDD. And what we aim is to coordinate better efforts and doing more in detailed cross-disorder, cross-tissue meta-analyses that could help us identify those biomarkers and potential drug targets for psychiatric disorders. And I want to thank my lab, mainly Sheila Namagatsu, who did perform a lot of the work that I presented today, collaborators at Mount Sinai from Yasmin's Hertz lab, and others, and funding agencies, of course. Thank you.
Thank you very much, Janitza. It's a lovely both overview and what's been done and what's to come, and some very exciting work ahead. Thank you for presenting all that to us. So I'm going to hand over to our third speaker of this session. Jens Hjerling-Leffler from the Karolinska Institutet in Sweden, and he's going to tell us all we ever needed to know about schizophrenia cells and circuits. So over to you, Jens. You're muted.
Now you can hear me, right?
Now we can hear you, yes.
Good.
Yeah.
Let's see if this works. Does that work right?
That works well. Great. Thank you. Take it away.
Thank you so much for this invitation. I'm really happy to come and present. So what I'm going to present today is one effort that you can use single cell data to try to make get biological understanding from GWAS data. And the title is a bit speculative, but the goal that we've had, or our approach to this, is to try to have a data-driven approach to understanding psychiatric biology, and with a focus on schizophrenia. And to do this, there's like kind of three main revenues where we can get unbiased or minimally biased data. One is, of course, understanding clinical heterogeneity. But since I am not a clinician, this is shrouded in a little bit of mystery to me. The other one is these kind of large-scale genetic studies, of course, and then studies of brain imaging, where we can image patients or controls or in different stages of disease. So one of the main efforts that I've been trying to get at is to move psychiatric research away from just having, or not just, away from having, relying on hypotheses that we are testing, and rather take a data-driven approach. So if you go through the literature, you can find almost any cell type in the brain being implicated in schizophrenia or in many psychiatric disorders. Ranges from different types of neurons to glia and non-neuronal cells, etc. So our approach to this was to see whether the GWAS signal actually could see if they were enriched in a cell type, but with the assumption that if a cell type expresses a lot of the risk genes, maybe it's underlying the etiology of the disorder. And this approach is a series of papers that we've done in mouse, and that was developed by Nathan Skeen and Julian Briard, working in my lab and in Patrick Sullivan's lab. This is a long-term collaboration between the two labs. And the whole idea is basically integrating GWAS and single-cell RNA-seq data to see if we can, that can guide us to understanding of where to look if you're a neuroscientist like me. So today I'm going to talk about this type of analysis, but in a new data set that came out late last fall. This is a single-cell data set from human brain. It's from 106 different dissections all over the brain, and it contains 3.3 million cells, and it's really the first time that we have a very large-scale single-cell data set that spans the human brain. And you can see in B here, hold on, let me see if this works. Yeah, so here you can see that if you do a single-cell sequencing of the many different parts of the brain, these are the main cell types that pops out. I think most of you have seen these UMaps or TS&E plots, which is a two-dimensional representation of the gene expression of each cell. Basically, there's a lot of caveats, but basically if two dots are close to each other, they are similar to each other, and each dot is a cell. And here we have Kimberly Siletti and Sten Linnarsson have color-coded this according to the clustering that a clustering algorithm gives rise to. And there's a dash here because these are the non-neuronal cells, and up here are the neurons. And I'm gonna mainly focus on the neurons because we've seen very little signal in the non-neuronal cells. The naming of these you should take with a grain of salt. It's very hard to do. So these are called median spiny neurons up here, for instance, but that's typically found in the caudate putamen. Here, on the other hand, are a lot of cells found outside the caudate putamen. This one is called splatter because of the way it looks like. It actually contains all the smaller subtypes like dopaminergic neurons, serotonergic neurons, hypothalamic cell types, etc. So the naming is hard to, it's very hard to name these data sets. And we have adopted the names that Siletti and et al. Put forward in their paper. Okay, so how do we do this? Well, first we do a cell enrichment analysis. And to do this, we first calculate the specificity of expression. And so we take all the genes expressed in the brain, and then we divide those into deciles. And then we look how much GWAS, how much heritability is in each decile. So this is an example of a median spiny neuron. We do this for each cell type, but here you can see in the beginning in green are all the genes that are shared between all cell types. Very low specificity score. So basically, this is these are all the genes that you need to be a cell. And then as you move up in specificity, you get into the reds and the oranges. These are now the genes that you need to make a neuron. So this is shared between all neurons, roughly half of the cells. And then as you go higher and higher up, and you go to the top part here, this is really the cells that define the function of this neuron. This is the genes that confers connectivity, specialized activity, molecules, etc. We call this the TDEP, and the top decile expression, and this is we argue now that this is where looking for enrichment, this top decile is really what defines the function and gives you an enrichment score for heritability. So we then use LD score regression to look how much signal there is in each of these deciles, and you can plot it like this. This is from our early study, and you can see that for schizophrenia GWAS, as we move up in the specificity for the median spinal neurons, we actually get more and more schizophrenia enrichment. If you contrast this to a GWAS for height, which we don't think is dependent on median spinal neurons in the caudate putamen, you can see that as we move up in specificity, we actually drop off. So we've done a lot of work trying to see what produces the most robust results, and we have landed on this approach that we are using LD score regression and looking at not the slope, but rather the absolute enrichment in the top decile in our method. Okay, so we have this method to calculate how much enrichment of heritability there is in the top decile for different cell types, and we marry it now with the human data set that was released. So you can do this. We here have a number of different GWASs divided into 5 major groups: psychiatric GWASs, brain traits, neurological, structural MRI, and control GWASs. And on the left side here, you can see all the superclusters, the major cell types of the brain. And if you calculate the enrichment, you can see that for the psychiatric disorders, we have a lot of signal for schizophrenia, bipolar, and major depression. And the blue line here it's neuronal cells above the blue line and non-neuronal cells below the blue line. You can see it's mainly neuronal enrichment. We actually have for brain traits very similar there are differences here, but this main cluster of cell types are the same that are enriched. For the neurological disorders, the only thing we find is epilepsy, where we see an enrichment for excitatory neurons. And in controls for BMI, we can actually find some enrichment in cortical neurons, which is or in excitatory neurons, which is in line with a central control of behavior. So now we can see we have all these cell types that are being enriched for different psychiatric disorders. And if we focus on schizophrenia, here on the left, we can ask the question whether these cell types independently enriched, or do they have a shared component? And to do this, we do a conditional analysis. So on the diagonal here, you can see the enrichment seen for this cell type in schizophrenia. And then the result is when we then compensate for different types of other superclusters, you can see that it can disappear. So for instance, for upper layer intratelocephalic neurons in the cortex, when we compensate for deep layer intratelocephalic neurons, that signal is actually gone. So that means that the signal between these two cell types is very much shared. On the other hand, all the other enrichments that we see remains pretty stable. And overall, I would say, except for thalamic excitatory neurons, it's a pretty robust signal, arguing that the most of the enrichment that we're seeing is actually specific to a cell type, which is maybe not that surprising from the TDAP analysis, but it could really very much not be the case. The other part we can do is look at subtypes of GWAS. So this is for major depression, and we can look at different subtypes of major depression and see that we actually get enrichment for different cell types across different subtypes. And we also get the more severe the GWAS or the subtype is, the more signal we're finding. So this was data on the supercluster level. In the selected data set, they've also identified clusters. So now we have, instead of 31 cell types, we can go down to 300 or 3,000. But we've done the analysis at the roughly 400 clusters. And if you do that, this is now for schizophrenia, you recognize the map of the brain cells, and now we can see where do we see the largest enrichment. In B here, we're just showing you the top cell types from A, and you can see that we have some really hot spots in amygdala excitatory neurons and hippocampal CA1. And actually, this is also amygdala, hippocampus, amygdala, and first here on the 7th position are we finding the first cortical cells. This was a bit surprising to us. But this is a kind of a map where we think that the major risk for attracting schizophrenia, perturbations, leads to a major risk for attracting schizophrenia. So, because this data set was came from 106 different dissections of different parts of the brain, we now have a score per cell type. We can now go back to the dissection and see how much did each of the cell types that we have on the map contribute to the data from that dissection. What this allows us to do is really to move from having just cell types to having individual brain regions in the brain. And you can see here are the different dissections from the 106 dissection from different brain regions. What you see in color here is the contribution of each of the cell types to that region. And by kind of summing up here, we can now calculate an enrichment score for each of the brain regions. And doing so, to keep this in reasonable time, we've color-coded the brain after risk. Here, the more red something is, the higher risk or the higher heritability for schizophrenia in a brain region. And two things really stand out. One is that the cortex is uniformly orange. There's nothing special about the prefrontal cortex that makes it special for schizophrenia. We know that prefrontal dysfunction is central for schizophrenia symptomatology, but we are arguing that this finding maybe shows or suggests that a connection model where the prefrontal cortex have specific connections is more important than the actual risk across the different cortical areas. And if you zoom in inside the brain, you'll see that the caudate putamen is actually not midbrain, and the thalamus are not that enriched, but we have a really hot spot here in the hippocampus and in the amygdala. So this is there's of course a large literature that has looked into the function of amygdala and hippocampus in schizophrenia, but I think this is the first time that we really have a truly data-driven way to prioritize using genetics and transcriptomics to prioritize brain regions. So on the right-hand side, to follow this up, we have a preliminary analysis. This is ongoing work a little bit. But we took fMRI data from patients and controls, an existing data set that we downloaded, and then we did something called a recursive feature elimination analysis. So what that is, that we feed it the entire network, and then we have a machine learning algorithm removing we teach the machine learning algorithm to tell the difference between cases and controls. And then we train the algorithm, and then we say which connection in the brain, functional connection, is the least necessary to do this. And then we remove that, and we retrain the algorithm, and then we remove the least important one in order to boil down into a core set of connections that are really essential for telling apart schizophrenia from patients from controls. And if you do so, our analysis show that, again, this is the core network that we find, and we can see that this is it highlights the role of highlights a role for the amygdala, the hippocampus, prefrontal cortex, and some other cortical regions. So we want to argue that these two kind of approaches converges on highlighting the connectivity between amygdala, hippocampus, and prefrontal cortex as a main culprit for or a main place to start looking for schizophrenia changes for neuroscientists. So if you're interested in this, we put it on medRxiv, so you can find it if you search for our names. And I'm really happy to have your input. If you read this, please, and you have ideas, please write us. We'd love to get input on this. So the conclusions then is that beyond cell typing, having quantitative measures of cellular diversity opens new frontiers. This is one example of a broader definition of functional genomics. We think that we can observe a convergence of different modalities of investigation, both cell type and circuit genetic risk enrichment analysis and functional connectivity highlights amygdala and hippocampal circuits. And we're really interested in seeing how this has a chance to how it correlates with clinical data. So I want to thank everyone who did this. Shuyang and Arvid from my lab did most of the work. Shuyang has since moved on and now started her own group. So keep an eye out for stuff coming from her lab. This is a very long time collaboration between my lab and the lab of Patrick Sullivan. The fMRI was done together with Johan Lundström, Fahim Adarki, Yue and Giovanni at the Gothenburg, and this is Karolinska, and then we've had statistical help from Naomi and her group at the QBI in Oxford. And with that, I'm happy to take the questions. Thank you.
Thank you very much, Jens. That was really interesting. I look forward to reading the preprint, which it's fabulous. So we now have about 8 minutes to the end of the hour for questions. So if you have questions for any of our speakers, you can drop those into the Q&A box, or if you just want to raise your hand, I can open the floor to you. So while people are thinking, maybe I could ask all three of you, what are you hoping to achieve in the next year that would really embed your group fully with the PGC, and how can we help you achieve that? Jens, maybe I can.
Yeah, so one of the things, so as Mary Ellen pointed out, we had kind of two criteria for starting projects in the group. One has been to get started, to find some low-hanging fruit that we can do. But the other criteria has been to try to build a resource that can be useful for the PGC. So basically, so those are the two kind of main criteria that we had for choosing. So if you have any ideas of what could be useful, we would love to hear about it, because this might be particular to different working groups. But if you have ideas of resources that you would like to see us to build, I think that would be very welcome feedback.
Okay, thank you. And I'm sure it'll be very valuable across the PGC. Mary Ellen, you touched on various areas in your introduction.
Yeah, and I suppose there's the conceptual drivers of our project choice, but we also just want to make sure we've got all our logistics smooth. We need to get an ethics application sorted for our collection of data. We want to get our patient involvement going. And it's a kind of iterative process of understanding where we should sit within this organization as we visit the different groups. We've learned so much from every group we've visited, and I think our plans are very much updated by that new information as it comes in.
Great. Yeah, thank you. And so I suppose that's also a call out for people who are listening currently or online, that if their working group hasn't already heard from you, you'd be welcome to further.
Yeah, it's just been ad hoc so far, whoever's invited us.
Yeah.
So please do get in contact.
Super. Thank you. Great. And Janitza, as the lead of an analysis group within the functional genomics, what would you like to add?
So it's a question if there's a lead analyst. I mean, we have identified different projects in the meta-EWAS working group. As Jens mentioned, we will start working with the low-hanging fruit, but also hopefully to fill out the gaps that there are different psychiatry disorders that haven't, like this kind of research hasn't been done yet, or not done at a larger scale. We have identified at least 3 different projects, and with the intention of having different analysts, so there's more commitment and opportunities for leading this work from members of our group. I did forget to mention that in addition to other PGC groups that have similar efforts are part of our membership to help coordinate efforts better. We also have representative from LAGC, from the Latin America Genomics Consortium, who also recently created their own group to improve representation of Latin American populations in this work. And also we'll be meeting with the PGC Africa working group, which we know they also have DNA methylation data. And hopefully we can also have representation from their group. And also looking at this from different ancestral groups as similar to GWAS. as well. But yeah, we will have multiple
Lovely.
analysts. There's no
Great.
like
Yes. Yeah. No. As you know,
project, yeah.
he was leading this project. Yeah, and it's been really nice to hear how this is all. You know, the ancestry part is just embedded in this from the start as well. Yay. So I've just asked Mary-Ellen just to share the site that she had for those of you who have listened to the talks and now realize, oh yeah, I can get involved here. So please do scan those and get further in touch. We have no open questions, which I think we should take as an absolute success that you that you covered everything beautifully clearly within the presentations, but people now know exactly how they can get in touch with you to talk further, and I hope you'll get many further contributors and people to work with. It's been a fantastic overview session of everything that you hope to do and plan to do over the next few years, and we're delighted to have you with us as the new PGC Functional Genomics group, and welcome to the family.
Thank you very much for having us.
Thank you.
Thank you so much.
So we'll finish there. Thank you everyone for joining this worldwide lab meeting. These happen periodically across the PGC. Please let me know if you have suggestions of areas that we should cover, or if you would like to offer a talk. It can be work related to the PGC or outside the PGC. Either are welcome, and all talks are posted on the PGC YouTube channel after live presentation. Thank you all for being here, and we'll finish there. Bye, everyone.
Bye.